My name is Erica Ramos, and I would love to welcome you to today's NSGC webinar on rare disease. What role do genetics play? We do encourage you to ask questions throughout the presentation. If you have any questions that you would like to ask our speaker, Kelly, please click on the question and answer tab and type a question into the chat box. All questions submitted will be read aloud during the question and answer portion of this presentation. And by submitting a question, you are consenting to have the information contained in the question read aloud during the presentation. Please do not submit any personal identifiers in your submission. As a courtesy to the presenter, all participant telephone lines have been muted automatically. Should you experience technical difficulties during this presentation, please use the question and answer tab to submit any questions or concerns our way. NSGC offers a wide range of educational programming. The information provided in this presentation is not a substitute for medical or professional care, and you should not use the information in place of a visit, call, consultation, or the advice of your physician, genetic counselor, or other healthcare provider. Now it's my pleasure to introduce Kelly East as our speaker for this webinar. Kelly is a certified genetic counselor and clinical applications lead at the Hudson Alpha Institute for Biotechnology in Huntsville, Alabama. At Hudson Alpha, Kelly oversees and participates in the provision of genetic and genomic counseling services for research projects and clinic, and leads the development of educational experiences and resources for healthcare providers and trainers and patients. Kelly is an adjunct faculty member for the University of Alabama in Huntsville College of Nursing, as well as the Edward Via College of Osteopathic Medicine. Thank you, Kelly.
Thank you, Erica, and thank you to NSGC for giving me the opportunity to speak with you tonight and share with you a little bit about rare disease and the role that genetics and genetic counselors can play in helping patients and families that are impacted by rare diseases. So let's jump right on in. When you think about rare diseases, they may affect just one person among thousands, but when you combine together all rare diseases, it can have a gigantic impact, affecting an estimated 30 million Americans. When I subtitled this presentation, When Hoofbeats Don't Mean Horses, I did that because when we talk about rare disease, we often use an analogy of a zebra. When we think of rare diseases like zebras that are unique, they're uncommon, sometimes they're difficult to find unless you're at a zoo. But when you see signs of them, like you hear horse beats coming, without close examination, you might be quick to mistake it for a more common explanation of hoofbeats, like a pack of horses. And the same can be true of rare diseases, that they can be unique and uncommon, they can be difficult to diagnose, and their symptoms are all too often mistaken for other more common conditions and explanations. So let's take a look at where we're headed in the next 30 minutes or so. This webinar is going to explore the topic of rare disease and the journey that patients can go on while they're trying to find an explanation for their symptoms, moving from the rare unknown to the rare known. And often this journey can involve genetic testing and meeting with genetic specialists like genetic counselors. During the webinar, we're going to discuss how getting a genetic diagnosis can impact a person's life, from medical care to their family, to their options for social support. And in contrast, we'll discuss what it means if genetic testing is performed and a diagnosis can't be made. And then lastly, we'll highlight some resources that are available to individuals that are impacted by rare disease, that are living both with and without a diagnosis. So let's first talk about what I mean when I say rare disease. That term, rare disease, is defined in the United States as a disease that affects fewer than 200,000 Americans at any given time. Over in Europe, they have a slightly different parameter, a disease that affects less than 1 in 2,000 people. I don't want to get too hung up here over that definition and what considers a rare disease and what's not a rare disease. But in general, a rare disease is exactly that. It's something that's rare. It's something that affects relatively few people, that most people may have never heard of or known someone affected by. And while a rare disease by itself may affect relatively few people, when you combine all the people that are impacted by the 7,000 known rare diseases, you have a lot of people. You've got 30 million Americans or so. And how many other rare diseases are still out there that have not yet been named or classified? So most rare diseases are caused by underlying genetic changes, And so oftentimes rare disease is a reason for genetic consultation or meeting with a genetic counselor and having genetic testing done. This doesn't, though, mean that most rare diseases are inherited from parents. We'll talk more later about the difference between genetic diseases and inherited diseases. Just to cover a few more statistics for you, 50% of rare diseases affect children, and in fact, 30% of children with rare diseases don't live to see their 5th birthday. And one thing that many rare diseases have in common is a generalized lack of scientific knowledge about the disease and the development of targeted treatments because they are rare and impact a relatively small number of people compared to much larger diseases. And in fact, 95% of rare diseases do not currently have a specific FDA-approved treatment, and about half of them do not have a specific foundation or advocacy group supporting and researching those diseases. So when genetic counselors find themselves meeting with families that are affected by rare disease, we often talk through with the family their unique journey. A journey that begins the moment that they realize that either themselves, their child, or a loved one has some set of symptoms. This could be parents that learn that their toddler is not meeting milestones, or a previously healthy adult that's now having difficulty with muscle control. Every patient or family's journey is different and unique to them, but one thing that most of them have in common is that they're full of curves and hills, and perhaps even an occasional U-turn. So when you're facing a suspected rare disease, one of the main goals is often getting a diagnosis and a definitive explanation for why symptoms exist and what a person is facing. A diagnosis may be able to be made simply based on a set of symptoms, but often genetic testing is done to help confirm a specific diagnosis. Not all rare diseases are genetic in cause, but many of them are. So it's very common for patients with rare diseases to undergo one or more genetic tests while they're on that journey to get a diagnosis. A patient may have a genetic test that turns out negative, meaning that it didn't find any genetic changes that are responsible for the disease or symptoms. And then a bit later down the path, their working diagnosis changes a little bit because of a new symptom that develops or an evaluation by a new doctor or specialist. And then maybe there's some more negative genetic tests along the way, some more new symptoms. And then eventually another genetic test that's ordered or a new test becomes available that provides that long-awaited positive result. And a diagnosis that is based on genetics. And in working with families with rare disease, this kind of path, this winding path with lots of kind of curves and curveballs that can be thrown your way is all too common. And for too many people that they're still on this journey and don't yet have that specific diagnosis. So if we think back to the slide with the kind of pathway on it and the hypothetical patient that we're talking through that's had all these genetic tests that have been negative before one comes back positive with that confirmed diagnosis, you might be wondering how can that be? If a person has a genetic diagnosis and has a genetic change, how can genetic tests ultimately be negative and fail to make that diagnosis? So to answer that question, let's take a little step back and let's talk about some basic genetic concepts and get all of us on the same page before we move forward in the presentation. A person's entire genetic makeup is called their genome. And you and everyone has a complete set of that information, their genome inside every one of their body cells. Your genetic information is made up of DNA that's twisted up very tightly into these structures called chromosomes. Humans have 46 chromosomes, or 23 pairs. And between all those chromosomes, they have about 6 billion letters of DNA. Those letters A, T, C, G, that you might remember from biology class. Now, genes are specific segments of DNA that provide a specific instruction for the body. For example, an insulin gene provides instructions to the body on how to create the insulin protein. And we have about 22,000 of those gene sequences within our DNA, as well as a whole lot of DNA that's not inside a specific gene at all. In fact, just about 1 to 2% of our overall DNA is found inside of a gene. The take-home message to all of these, this complicated terminology and numbers is that the human genome is a really big place with literally 6 billion places that a genetic change can occur. Genetic changes can vary widely in size. Some genetic changes can be very, very large and involve whole chromosomes or big sections of chromosomes. This picture here is from a type of genetic test called a karyotype, which is quite literally a picture of a person's chromosomes. Karyotypes are useful in detecting genetic conditions that are caused by having extra or missing whole chromosomes or large sections of chromosomes. This specific example has an extra copy of chromosome 21, which would indicate a Down syndrome diagnosis for this individual. So this kind of genetic testing is really great for those kind of conditions, but it's not ideal at all for detecting smaller genetic changes that are at the level of the DNA sequence. So the type of test depicted on this next screen is what we call a microarray, and its goal is to look for sections of chromosomes that may be extra or missing, but smaller than what you can see on a karyotype. The boxes on the screen indicate specific chromosomes, and where the line deviates from the middle indicates places where there may be a section of DNA that might be added or missing. This kind of test essentially zooms in on the chromosomes and lets you see smaller changes than what you can see on that karyotype alone. But it still has limitations and is still not able to look for very small genetic changes that might involve only one or a few letters of DNA. I mentioned earlier that the human genome is ultimately made up of 6 billion DNA letters, and a person can have a genetic change at any one of those places. So to find a small genetic change, different kinds of genetic tests have to be ordered. DNA sequence-based tests can actually read the DNA letters and look for differences in them. A difference could be a switch of a single letter. So instead of having an A at a specific location, a person has a letter C instead. Or a change could be one or a few letters that are either extra or missing. DNA sequencing tests are able to find these kind of changes. But there's a whole variety even of DNA sequence-based tests that vary in the amount of the DNA that they can explore. You could have a test that looks only at one letter of DNA, or a single gene, or several genes, or even the whole human genome. So if we compare the different types of genetic tests that are currently out there available that can be used to help make a genetic diagnosis for patients who might have a rare disease, we'll see that they all have varying benefits and limitations. We can think of tests in terms of the level of coverage, so how much of a person's genetic makeup is getting looked at, while the resolution of a genetic test looks at the level of detail that the test is looking at the DNA. So something like a karyotype, that picture of the chromosomes, has wide coverage because it looks at all the chromosomes. But it is relatively zoomed out, so it has very poor resolution. Other DNA tests the DNA-based tests are very targeted. They can look specifically, they have very high resolution because they're looking at the DNA letters, but some of them might be very targeted, only looking at a single spot in your genome, but others are very big and can look at lots and lots of the DNA at one time. So things like exome and genome sequencing that are kind of in the top right corner of the slide have both very high levels of coverage as well as high levels of resolution. If the term exome isn't familiar to you, what we mean when we say exome is that is the set of all of a person's genes, so the DNA that is inside all of those gene sequences, but it does not include the DNA that would be in between those genes. So that's really that 1% to 2% of our DNA that is the instruction manual for proteins in our body. Whole genome sequencing is really the biggest genetic test you can possibly do that has both very high coverage, very high resolution, is looking at all of the DNA, but even with whole genome sequencing, you're not able to detect at this point 100% of genetic changes. So because of that, you can imagine that it's possible for a genetic change to be present in a patient, but because of the specific test that's used and the limitations of it, the laboratory isn't able to find it. So another reason why a genetic test could be negative, even when there is a genetic change present, is if the laboratory, even if they see a change, the testing laboratory may not be able to fully understand the impacts of that genetic change and determine that it is indeed a cause for symptoms. And this is because not all genetic changes cause problems. In fact, most genetic changes that occur do not have any negative impacts at all on health or disease risk. So as laboratories look for and find genetic changes in patient samples, they have to evaluate the change to determine whether or not it's actually the cause of disease, something that we will call pathogenicity. So consider one of these picture games that you may have played before in either newspapers or magazines, where you've got two images with a set of differences in between them. As soon as these pictures went up on the screen, you probably started picking out the differences. The picture on the left has a green flag, while the picture on the right has a red flag, which probably has something to do with the extra waves in the ocean or that possible shark fin sticking up. That's a pretty major difference that would have a profound impact on your day at the beach, as would whether you forgot your sunscreen, like in the picture on the left. Other differences between these pictures are much more benign and don't really have a functional impact on your beach day. Things like the color of the boombox or the umbrella. But now let's pay attention a little bit to that crab that's crawling in the sand. Its coloring is slightly different between the two pictures. Do you think that would be an important difference? Maybe, or maybe not. We might need some more information before we can decide. Would one specific color of crab indicate a more dangerous species? That one's a little bit harder to determine from just glancing at it. And the same thing is true of genetic differences. When a lab looks at a patient's DNA sequence and finds differences from what they would expect, they have to do research and determine whether or not it would cause a problem. And so typically genetic laboratories will classify genetic changes on a scale from 1 to 5, where at one end of the scale you have benign genetic changes, meaning a change is definitely not going to be causing a disease. This is where the vast majority of genetic changes are, and changes like these don't usually even get reported by a testing laboratory back to doctors and patients because they are known to be not disease causing. It's really important to keep in mind that having a genetic change is not inherently a bad thing. In fact, every person walking around has millions of genetic changes inside of their genome. At the other end of the scale, though, we have what we call pathogenic variants. And these are variants that have strong evidence that they do indeed cause disease. These kinds of genetic changes can diagnose a person with a disease or tell a person that they have a high risk of disease in the future. And then there's several levels right in the middle of the scale that have differing levels of uncertainty attached to them. There's variants that are likely benign or likely nothing to worry about, or variants that are likely pathogenic, meaning they look like they quite possibly are a diagnosis or a cause for disease, but there's not enough evidence to say that with 100% certainty. And the middle of the scale is truly uncertain. Variants right there are usually called variants of uncertain significance because there's just not enough evidence to really make a call one way or the other. We typically will consider these variants of uncertain significance innocent until proven guilty. And over time, as more is learned, these variants will become, that are uncertain now, will become more clear and will start pushing towards pathogenic or benign. So you can see from these slides and this presentation that there's a number of reasons why a person could have a genetic condition, but ultimately have a negative genetic test result. It could be because the genetic change is there, but that the laboratory isn't able to find it because of the testing that was used and the limitations of the technology. Or you could have a variant that was there that was found, but that the laboratory didn't have enough information to make a call and to report that as the diagnosis because of limitations with our understanding and our knowledge of genetics. It's also possible, though, that maybe a patient has a set of symptoms that are due to genetics, but it's not a single genetic factor that's totally controlling the symptoms. Maybe there's multiple genetic factors that are working together to create symptoms with or without maybe some environmental puzzle pieces as well. These complicated puzzles that we call complex genetic disorders are still incredibly hard to put the puzzle together and to figure out at this point in time. And then lastly, it's also possible that someone can have a negative genetic test result because their symptoms are not actually genetic in cause at all. But it's easy, I think, to sometimes put too much confidence in getting a negative test result and saying that means that a condition is not genetic, because when you're dealing with rare disease, most of them are genetic. And so in a lot of cases, ultimately there is an underlying genetic cause and genetic change that isn't able to be identified yet. So I want to show you just a little bit of data from my own institution here at Hudson Alpha, which is a genomic research institute in Huntsville, Alabama. We've been conducting a study for the last several years now through funding from the NIH to offer whole genome sequencing, so that really big genetic test for families that are affected with unexplained developmental delays. We look at the affected individuals that have developmental delay. We look at their entire genetic sequence to try to find an underlying genetic cause. And we call these primary findings because they relate to the primary reason that we're doing the test in the first place. And so you can see from the graph that I'm going to put up on the screen that about a quarter, or 25% of cases, we were able to find a definitive cause for their developmental delay. So they got back a pathogenic or a likely pathogenic result. In another 19%, we found one of those kind of pesky, uncertain variants that may or may not end up being the actual cause, but that we do actually report back to the family so that it can be watched over time and continue to be studied. And then in a little over half of the cases, we get back that negative result, meaning that we weren't able to find any possible genetic cause for developmental delay at this point. In many of these cases, we're still quite suspicious when they're negative of a genetic cause, but likely our technology or our knowledge is keeping us from being able to find it. We'll talk in just a little bit about how Hudson Alpha is continuing to look through those cases and try to continue to find diagnoses in the ones that we aren't able to find one in initially. But many of the families that we enroll in the study have had lots of genetic testing done before they come to us and enroll in this study. So the data that we've got here really speaks to the power of whole genome sequencing and the fact that we're able to get a diagnosis in 20 to 30% of these really challenging cases. It's also worth mentioning, though, that when you do big genetic tests, like whole genome sequencing for some reason, that it's possible to find what it can be referred to as secondary findings. These are genetic variants that are found in a person's DNA that's not related to the reason that the test was done. For example, in this study, so we sometimes, not overwhelmingly often, but sometimes we'll find genetic variants that are associated with a high risk of cancer or heart disease, or the fact that an individual is a carrier for cystic fibrosis or sickle cell anemia. These kinds of findings have no bearing on why a person has developmental delay or neurologic illness, but they still could be quite useful to that person, to their family, and to their medical care going forward. So we've been talking up until this point about getting a diagnosis by a genetic test, and I do want to acknowledge that that's not the only way to achieve a diagnosis. That some diagnoses can be made by looking at physical symptoms, or they can be made based on the results of a test. Symptom-based diagnoses, though, are things like cerebral palsy or autism spectrum disorder or epilepsy, things that tell you about the symptoms and the medical needs of a particular person, but often do not tell you the answer of why those symptoms exist. The cause or explanation could be a number of things like physical accidents, premature births, a number of other external causes, or they could be due to an underlying genetic condition. And in the research study I mentioned on the previous slide, we actually enroll a number of patients who have these more symptom-based diagnoses like CP or autism or seizures, but these symptoms are likely due to genetics in a lot of cases. And really the purpose of genetic testing is to make a genetic-based diagnosis and explain symptoms. So getting a diagnosis, there's lots of reasons why a patient, a family, a healthcare provider would want to continue to search for a genetic-based diagnosis. Getting a diagnosis may not mean that there's going to be a new available treatment that's going to suddenly cure a condition or take away all the symptoms. While there's exceptions, most often when you're making a genetic diagnosis, it's not going to significantly change the disease course or improve the prognosis necessarily in a patient. But getting a diagnosis can change how healthcare providers are caring for that patient. And sometimes, for example, this can help providers pick the best treatment out of a set of options, or avoid treatments that are going to be known, that are known to be harmful or are completely ineffective for a particular group of patients with a specific disorder. Or getting a diagnosis can help provide those patients with the best prognosis information that we can, and give families and patients a better view of what their future may hold. Other more medical values of getting a genetic diagnosis can be helping to suggest any additional screening that might not have been originally thought about. For example, if a patient is being monitored for seizures and gets a genetic diagnosis, it's possible that the particular condition that they're getting diagnosed with might also be associated with a high risk of heart problems, and that would lead the medical team to suggest regular cardiac evaluations. Or it's possible that maybe there are clinical trials or research studies that are tied to a specific genetic condition that a family can get involved with once they have a diagnosis. This can allow families to help contribute to that scientific knowledge and what we know about diseases, which is something that is often very lacking when you're dealing with rare diseases where we don't know as much. And lastly, knowing why a disease has occurred in a family can help us answer the question of whether it's going to occur again, whether it's the parents of a child that have a rare disease, their siblings, or the person with the rare disease themselves looking at their own childbearing. Because sometimes it might be easy to assume that if a person has a genetic disease, that it's definitely going to happen again in a family, or that the chance is very high. And often that's not the case at all. And so you may hear people use the term genetic disease or inherited disease interchangeably when actually they don't mean the exact same thing. So let me explain. A genetic disease is a disease or condition that's caused by a genetic change in a person's genetic makeup, plain and simple. This genetic change may or may not have been inherited from a parent. So how is this possible? There are new genetic changes that occur in every single person and every new generation. So each one of you listening to this presentation tonight has a number of genetic changes in your DNA that you do not share with your mother or father. Sometimes these new changes can cause a genetic disease. And in contrast, an inherited disease would be a disease or condition that's caused by a genetic change that we know was indeed inherited from a parent. And so determining whether or not a disease-causing genetic change has been inherited from a parent is really critical when we're trying to answer the questions for recurrence for a couple and their families. So those are a lot of the more medical reasons for searching for a diagnosis, but there's a lot of non-medical reasons as well. From a personal standpoint, getting a diagnosis can end that part of the diagnostic journey. In some ways, getting a diagnosis begins a whole new journey for a patient and their family. It provides an answer to the question of why. And kind of tied in with that, it can help address some potential parental guilt and blame that may exist. Even though as a genetic counselor, we talk to parents at length about how nothing they did or didn't do has caused a child's symptoms, we find that until we're able to really give a definite diagnosis and answer that why, it can often be hard to put aside some of that guilt and blame. And in addition to connecting families to research studies and clinical trials, getting a diagnosis and having a genetic name for a set of symptoms can help connect families with support groups and advocacy organizations that are related to a specific diagnosis. Families are able to then meet each other either in person or virtually, families that are on a similar journey, and that they can share encouragement with and advice and troubleshooting and be a network for each other. With online websites and social media, this task is getting easier and easier, which is great that we can make these connections, because when you're dealing with rare diseases, quite often the next closest person that has the same disorder may be across the country or across the globe. And so when we walk through the benefits of getting a diagnosis, both from the medical perspective and the personal perspective, getting a diagnosis is really important to a lot of patients and families and healthcare providers. But what happens if a family is kind of stuck in that diagnostic journey, that genetic tests have been negative and that a diagnosis hasn't yet been made? And what happens in those situations? So one thing that might happen is a doctor or a genetic counselor might suggest some additional genetic testing to be done, either at that time or waiting in the future when new and better testing becomes available. We know that genetic testing technologies are going to evolve over time and get better, and so there may be additional testing that's recommended down the road. Or alternatively, a doctor or genetic counselor might suggest that the data from a genetic test that has already been done be re-looked at or reanalyzed by a laboratory. And this speaks to the fact that our understanding and our knowledge of genetics is going to get better over time. And looking at even the same data in the future might allow for a diagnosis to be made that couldn't be previously understood. But both of these options require that patients that they stay plugged in and stay connected to their genetic healthcare team. And so we always encourage patients, even if they don't get a diagnosis and a genetic test is negative, that they check in with their genetic healthcare team periodically to see what any new recommendations may be. And so going back to that study that I mentioned earlier here at Hudson Alpha, I want to show you how we've been going back and looking at patient data over time as the scientific community learns more about genetics and its role in rare disease, how we're able to look at patient data in a new way and ultimately find things that we weren't able to find the first time around. And so if we look at patients that we have given results back to in that developmental delay study, we threw up this table, and there's a lot of information in this table, but let me unpack this for you just a little bit. So this shows you at the point where we pulled this data, we had 359 cases, so families affected with developmental delay. And this shows you how we initially called these patients, or what kind of results we gave back to them, whether it was a pathogenic variant, a likely pathogenic, or VUS is that variant of uncertain significance, or none are the cases that we weren't able to give any, we weren't able to find any genetic changes that were reportable back to the family. And this shows how over time, whether it be a couple months or a year or two down the road, how getting new information has allowed us to either find a variant that we initially didn't report on at all, or change how we classified or understand a variant to be involved in disease. So we had 13 cases there where we went from a negative result where no diagnosis was made to finding a genetic change when we re-looked at the data that we felt should be reported to the doctor and the patient. Just above that, there were 5 cases where we had a variant that was initially called out as uncertain, where we had a variant that we didn't know what it meant, but with new information, we're now feeling more confident that that variant is indeed the diagnosis for the patient. And then on the flip side, in green in that last column, you can see that there are some variants that we initially, that got flagged that we thought might be a diagnosis, and with new information, we're actually feel like those variants are less likely to be the diagnosis and are more likely to be a benign variant that is not related to disease at all. But what this data really highlights is the value of staying in touch with a healthcare team, staying in touch with your geneticists, genetic counselors, that just because genetic tests may have been negative thus far, that doesn't mean that a genetic diagnosis isn't going to come over time. So we just walked through a ton of information. And so before I stop talking at you and we start kind of working together through any questions that you have, I want to just highlight for you some resources that you might find useful if you are impacted in some way by rare disease, whether as a patient yourself or as a caretaker, relative, or friend of someone with a rare disease. So for individuals who may still be on that diagnostic journey, that's still looking for a potential genetic diagnosis for your rare or your unique symptoms, I want you to know about that there are support organizations that are there, that exist for the rare disease community more broadly, that are not tied to a specific condition, but are there to help advocate for and to support individuals that have rare disease, that are either diagnosed or undiagnosed. I also hope that through the course of this webinar and this presentation that you see the impact that genetic testing has in working through getting a diagnosis for patients with rare disease, and how genetic counselors can play a role as well. And so if you find yourself not already plugged in with a geneticist, genetic counselor, you can go visit this website right here, that's findageneticcounselor.com, to put in your location, and it'll help you find a genetic professional that's near you that can help you navigate this process of looking for a diagnosis and helping you evaluate a case and determine whether or not genetic evaluation or genetic testing might be of use. And my recommendations are largely similar for people who are at the end of that diagnostic journey but are still on a journey with rare disease that may have a specific diagnosis. The broad, more rare disease support groups are still a great resource for individuals with known genetic disorders, as well as a whole host of specific support groups that are tied to a certain genetic condition. I listed a few of them up here on the screen, but there are literally thousands more. But I encourage you to seek out a support organization that is tied to a specific rare disease of interest to you, and if you don't have one, to work with your genetic counselors and have them help you identify some avenues of support. And if there truly isn't one out there, perhaps by connecting with a few folks who have that do have the same condition, maybe that will be the beginning of a new one. I mentioned earlier for people who have a known genetic disorder that one of the benefits there is being able to look for and potentially plug into existing clinical trials or research studies about a specific disorder. Clinicaltrials.gov is kind of the centralized resource for searching all the approved clinical trials that are going on. So you can search this site for a specific disease or condition or a specific gene and see if there are any trials going on and what the status of those trials are. And even once a diagnosis is made, we still encourage patients and families to stay in contact with their genetic healthcare team. That geneticists and genetic counselors continue to help families across a lifetime with a rare disease, with providing information about conditions, recurrence within a family, treatment options, and helping make those connections with support groups, research studies, and trials. And ultimately, if you have questions, reach out to your genetic team. That genetic counselors, our goal is to be here for families and help support them and answer any questions that come up. So with that in mind, now let me do just that. Let's take some time, and I'm more than happy to answer any questions that you may have, or help clarify anything that's been covered in this webinar.
Great. Thank you so much, Kelly. This was incredibly helpful and informative. One question that I did have around keeping in touch with your genetics team, do you have any advice on how often patients and families should contact their genetic counselor?
Yeah, that's a great question. I think there's a lot of nuances from a particular case that would probably impact what that time interval is. So I don't want to say that there's a specific prescriptive time that should be the how long it should be before you check back in. I would probably look to your genetics team for that answer and see what they think is going to be the most valuable in that particular situation. I see ranges from anywhere from, you know, a few months if there's some really critical thing going on in a patient's journey to every year or every other year. A question that we get asked a fair amount has to do with people who have genetic testing that comes back negative, and kind of how often does it make sense to go back and have labs say, look at the data again, and do that reanalysis step to see if there are any new ways to look at the data and whether a diagnosis can be found that was missed previously. And again, I don't think there's not a set time. The data that we're starting to see shows that maybe somewhere in the interval of 18 months or so, or 2 years is a reasonable amount of time to give science a good set of time to improve our knowledge base. But again, there's no perfect answer that's going to work for everybody.
Great, thank you. And there's a question on insurance. Is insurance something that will typically cover some genetic testing in cases of rare disease?
Yeah, that's a great question as well. And also one that does not have a single answer that's going to apply to all cases. It is going to depend on the particular set of symptoms and the question that's trying to be answered and the type of testing that's getting ordered. So as you can imagine, when new types of tests come online and are brand new and available to doctors and genetic counselors to order, oftentimes brand new tests are not initially something that is covered widely by health insurance companies. But over time, as evidence grows that a test is really useful in getting diagnoses and providing answers, those tests do eventually become standard of care. And so there's a whole host of genetic tests that are often covered by insurance companies in a diagnostic space, where you have a patient that you suspect has a genetic disorder, and you're trying to find what it is. A lot of the things like the karyotypes and microarrays and even sequence-based tests that look at one gene or a set of genes. Those really big genetic tests, exome sequencing, genome sequencing, are much newer tests on the market, and you're going to see a lot more variability in when those things are covered by insurance and when they're not. But it's a changing landscape that if you ask the question today and ask it next year, there may be a very different answer, because we really are on the cusp of that being much more standard of care.
Perfect, thank you. Well, Kelly, this has been incredibly informative, and I want to thank you and all of the people who have taken time out of their nights to call in tonight. I did want to let everybody know that the recording will be shared and posted online. So if you do have questions or want to go back and get some more information out of Kelly's great talk, you can certainly do that, and that information will be on our genetic counselor webpage. Thank you so much.
Thank you.